Having a problem with your crops caused by pests and other harmful substances that may affect your crop yield? There is a solution to that problem by applying genetic modification. And what is genetic modification? Genetic modification is a technology that involves inserting DNA into the genome of an organism. To produce a genetically modified plant, new DNA is transferred into the plant cells. Usually, the cells are then grown in tissue culture where they developed in plants. The seeds produced by these plants will inherit the new DNA. The characteristics of all living organisms are determined by their genetic makeup and its interaction with the environment. The genetic makeup of an organism is its genome, which in all plants and animals is made of DNA. The genome contains genes, the regions of DNA that usually carry the instructions for making proteins. It is these proteins that give the plants its characteristics, such as the color of flowers and the height of a certain plant. So, how is it done? The first stage in making a genetically modified plant requires transfer of DNA into a plant cell. One of the methods used to transfer DNA is to coat the surface of small metal particles with the relevant DNA fragment and bombard the particles into the plant cells. This method is called the particle gun method. Another method is to use a bacterium or virus. There are many viruses and bacteria that transfer their DNA into a host cell as a normal part of their life cycle. For genetically modified plants, the bacterium most frequently used called Agrobacterium tumefaciens. The gene of interest is transferred into the bacterium, and the bacterial cells then transfer the new DNA to the genome of plant cells. The plant cells that have successfully taken up the DNA are then grown to create a new plant, carrying the new traits such as increased pest resistance, larger fruits, taller or shorter size of crops, and more.